National Advisory Eye Council, 6700B Rockledge Drive Conference Center, National Eye Institute, NIH, October 4, 2019. Strabismus, Amblyopia, and Visual Processing, SAVP programs. Perception and Psychophysics, Myopia and Refractive Error, presented by Sherry Wiggs, PhD.
All right, so as foreshadowed, I'm going to be covering the last two sections of SAVP. A Perception and Psychophysics program is really about vision functioning as it should. The myopia and refractive error is vision that's a bit disordered.
Strabismus, Amblyopia, and Visual Processing, SAVP. Development and Regeneration, directed by Tom Greenwell, PhD. Central Visual Processing, directed by Martha Flanders, PhD. Ocular Motor Systems, Strabismus and Amblyopia, and Sensory Neuro-Ophthalmological Disorders, directed by Humam Araj, PhD. And highlighted in a yellow box, Perception and Psychophysics, and Myopia and Refractive Error, directed by Sherry Wiggs, PhD. Thank you. Perception and Psychophysics. On left, showing 1860 photo of Gustav Fechner. Stimulus, arrow pointing to mind/mental processes, arrow pointing to percept.
All right, so the field of psychophysics has a pretty long history. You can go back to the mid-1800s with the German scientist and philosopher Gustav Fechner. He asserted that the mental processes that we use to interpret information from the physical world is susceptible to measurement. Now, that was actually a pretty bold idea at the time, but he had to put his career on the line to show that it could be done. And as a result, he founded the field of psychophysics.
On the right, psychophysics quantitatively investigates the relationship between physical stimuli and the perceptions they affect, labeled psychometric function for a yes-no paradigm.
There are fundamental quantitative methods that you use to be sure that you can titrate the relationship between the physical stimuli and the perceptions they affect. And so just as an illustration, I'm bringing up a well-known psychophysical function that Fechner worked on. That is the absolute detection of light. So you can see on the graph that along the x-axis, you can incrementally increase the intensity of a stimulus and then simultaneously plot on the y-graph the perception that resulted.
New slide labeled Perception and Psychophysics. On left, light gray box with 4 circular patches of blurry black and white lines.
And so this strategy of tight experimental control of the physical stimuli connected to the functional experience of perception continues to this day. And so stimuli such as these sinusoidal gratings or Gabor patches are in widespread use. And they're really appealing for a number of reasons. One is, as you can see, they're relatively simple stimuli, but they have a number of ways that they can be varied experimentally, either by the frequency or the orientation of gratings. They can be characterized mathematically pretty easily, so that's actually very useful for computational modeling. And we know that cells in the visual cortex just eat them up. So that's really, really useful for neuroscience programs. But the fact of the matter is, the world looks more like this.
On the right, a busy scene from a New York street with taxis, pedestrians, cyclists, street signs, and colorful storefronts.
And so it's cluttered, it's complex, it's colorful, it's meaningful, it's dynamic. It's all of those things. But people have been really interested to see if it is possible to study these without sacrificing that experimental and computational rigor that psychophysics brings. And so I'm going to highlight 3 different labs that are doing just that.
Natural scene processing. Natural stereo image with ground truth distance data. An image showing 2 men on the right. Labeled on the photo are an SLR camera directly in front of them, a laser range scanner below the camera, and a robotic gantry resting on the ground at their feet. In front of the camera setup is a scene with a building, trees, and a lamppost. References: Burge J., McCann BC, Geisler WS, 2016, Estimating 3D Tilt from Local Image Cues in Natural Scenes, JViz, Volume 16, page 2. Iyer AV and Burge J, 2018, Depth Variation and Stereo Processing Tasks in Natural Scenes, Journal of Vision, Volume 18, Issue 6, pages 4 and 1 through 22.
And the first one is Johannes Berg at University of Pennsylvania. He's an early stage investigator that we just recently funded. And he combines computational modeling and psychophysics to understand how it is that we estimate depth from natural scenes. And so in developing his algorithms, he recognized that it would actually be really useful to find out if the estimates are accurate. And so he developed what he refers to as a Frankensteinian device to collect these images with this data. It has a digital SLR camera set atop a laser range scanner set atop a robotic gantry, and the gantry continuously aligns the scanner and the camera so they are getting their data, that is the images and the distance data, simultaneously in the right places. And so just to give you an example of the templates that result, he can get these low noise, high resolution stereo images from the camera. And then those are templated and aligned with these co-registered laser measured distance data.
On the right, a series of black and white images. On top, labeled low noise, high resolution stereo images. Below, labeled co-registered laser measured distance data.
So he's actually collected the full data set. And what I want to highlight here is that, yes, he's starting to use them in his own studies on depth estimation, but importantly has put this data set out. It's available to the scientific community. And it's not just psychophysics labs that have contacted him to use these data. He's also heard from neuroscience labs and machine learning researchers. So
red text, reading database at http://natural-scenes.cps.utexas.edu/db.shtml.
So the NEI funds that went toward this are actually being distributed not just in his lab, but beyond.
New slide with a photo at top center showing kitchen cabinets below a window with trees beyond. Light from the window reflects off a corner wall. Items clutter the cabinets and counter. Reference, Henderson J.M. and Hayes T.R. 2017. Meaning-based guidance of attention in scenes as revealed by meaning maps. Nature Human Behavior, Volume 1, pages 743 through 747.
Okay, so another example of how we process natural scenes is I can show you a real-world image like this. And if I had an eye tracker getting data from all of you, I'd get something that looks like this.
Image at top left, same as image at center, but with blue scattered over the counter and cabinets, labeled Scene Fixations. Below, a heat map showing several areas highlighted in red matching the blue on the photo above, labeled Attention Map. Attention maps, empirically determined from viewer fixations.
So you can actually empirically obtain where your fixations landed, and then I can take those data, well, I can't, but people are much more mathematically inclined, can take those data and actually transform them into an attention heat map. In other words, those hot spots indicate where fixations landed more frequently or stayed on the image more. And that gives you a sense of how people, when looking at a scene, prioritized which regions that they wanted to look at. But these are just the empirical data of where your eyes landed. The question is why? Why are they going to certain places?
New heat map below the center image with red highlights on the area of the light reflecting off the wall and a bit on the window, labeled Saliency Map. Saliency maps, created by evaluating the stimuli on color, orientation, and brightness, Itty and Koch.
So one theory is that it's all driven by low-level features, such as color, orientation, and brightness. And Laurent Petit actually developed a model to test this. He could take these visual images, get the mathematical properties of where most of those low-level features were spatially, and then could create a heat map that he could then compare to see how predictive it was of the empirical attention map data.
Upper right, 4 small portions of the image, including bits of wall, curtain, and objects from the counter, labeled Examples of Low and High Meaning. Below, a heat map labeled Meaning Map with greatest focus on the object sitting on the counter. Meaning maps represent spatial distribution of scene semantics via crowdsourced ratings.
All right, but John Henderson at UC Davis said, well, I disagree. I don't think it's actually low-level features that are directing our visual guidance, but it's actually the high-level semantic content that's in the scenes. And so he used a procedure very similar to ET's in terms of developing the salience maps for low-level features, but did that with semantics. And so now we have two models here, the low-level feature-driven visual attention model and the meaning map model for high-level semantic. And I know I've glossed over the methods so that it's almost like I'm saying, and some magic occurred here to make these maps mathematically viable. I'm happy to answer questions about those later, but in the interest of time, I'm going to say that the home point, the important one, is that the salience map and the meaning map can then be tested against each other to see which one is more predictive of the actual empirical fixation data. And if you have just a free viewing episode like this, where you're just shown the image and you look, the meaning map is actually more predictive. But one could say, well, wait, maybe we're biased to do that. And so...
New slide, brightness search. On the left, labeled fixation, a gray rectangle with a single red plus in the middle. In center, labeled scene viewing, a photo of a chaotic room with tables and boxes and other items. On right, labeled response, a gray rectangle with a smaller box inside framing a small O. Overwritten, how many bright patches did you count? Below the images, an arrow pointing from fixation past scene viewing to response labeled times 20 scenes. Reference, Peacock CE, Hayes TR, and Henderson JM, and Hayes TR 2019. Meaning guides attention during scene viewing, even when it is irrelevant. Attention, Perception, and Psychophysics, Volume 81, Issue 1, pages 20 through 34.
In a recent study that Henderson's lab just published this year, they pushed the subjects to treat it like a low-level feature experience. So they instructed them to just search for how many bright patches they could see in the image.
Salience explained 1% of the overall variance in attention, meaning accounted for 18% of the overall variance in attention. Bottom right of slide, a graph showing unique correlations versus scene number with dots for image seen. Bar graph shows that meaning is significantly higher than saliency on average.
And meaning still won out. So the meaning maps were accounted for the overall variance better than the low-level feature ones, and the individual correlations also mapped out better. I will say, again, in terms of a really innovative, unique method that was developed with this NEI funding, is not just staying in John's lab, but his colleague Lisa Oakes, also at UC Davis, who's studying how visual processes develop, is using the same method in 4, 8, and 12 months old. What she says, the period of time where children transition from learning to look to looking to learn. And it's going to be really interesting. She's just starting this data collection, so it's going to be really interesting to see if these semantics come online to a certain point of time in development.
Active vision and real-world environments. Images top left showing hikers on paths through woods on flat, smooth dirt, medium, rough dirt and small rocks, and rough, large rocks, terrain. Below those, image of a stick figure person over a purple background, with yellow and green hotspots showing previous foot placements, and a pink line pointing in front of the person towards additional hotspots. To right, image of rocks from walker's point of view, with a freeze frame of an image of the person's eye at the upper right quadrant. Below, series of graphs labeled look ahead versus time, 2D gaze on ground, and eye in head position. To right of images, how do walkers gather the visual information needed to guide foot placement on natural terrain? Mobile eye tracker, infrared blocking face mask, motion shadow IMU-based motion capture system, records gaze and full body kinematics during locomotion over various terrains. Tuning gaze to environmental demands. Real-world motion patterns. Reference, Mathis, Yates, and Hayhoe, 2018. Gaze and the control of foot placement when walking in natural terrain. Current Biology, Volume 28, pages 1224 through 1233.
Okay, so the last lab that I want to share is not just natural scenes, but putting you into nature for the visual experience. So Mary Hayhoe at the University of Texas at Austin and her postdoc, John Mathis, who was a K-99 recipient, and yeah, yay, Neeraj. And he just transitioned to his R00 at Northeastern. They developed this remarkable data collection system to see how it is that walkers gather visual information that they need to stay upright as they locomote through various terrains, including a pretty rough terrain. And it combines a mobile eye tracker and a motion caption system, so you can record the person's gaze and their full body kinematics as they are walking over these terrains. And it's really a great opportunity to look at unique data, such as how we tune our gaze to different environmental demands, or how real-world motion patterns are experienced, unlike in the lab. So,
New slide with movies of stick figure and walker's view, as well as graphs below, labeled DAG_2016-01-20_take0005, video courtesy of Mathis and Hayhoe. Stick figure video shows foot placements and eye focus highlighted. Video of eye tracking shows fixations on a particular point followed by quick eye movements to a new location. Graphs below track eye and head movements over time.
I want to give you a sense of what this data collection looks like in real time. It's multiple streams of data as the person is walking. On the left, you can see a reconstructed skeleton with the footholds, past, present, and future shown in the dots. The pink line is the gaze vector, where they're looking as they're walking. And then on the right, you can see a frame from the eye tracker. It's showing where the eye is in the head, and then the blue crosshairs are showing the person's 2D point of regard as they're moving over rough terrain. And I don't think anybody fell during the experiment, although I would have.
New video showing retina-centered video to left, which indicates what image would be recorded by the retina, and head-centered video to right with the scene as tracked by the head camera. Both motions are displayed on graphs versus time below the videos. Stick figure is at bottom left. Video courtesy of Mathis and Hayhoe. Video of the walker's eyes are shown at the bottom of the head center video.
So the next one is really interesting. So what they've done is there's a number of ways they can pull these data. And what they did here is to look at the retina-centered vision. In other words, how the motion patterns present on the retina as you're walking. And what you will probably notice is that it is not a smooth, nonstop velocity, but there's a lot of acceleration and deceleration. And this goes counter to what the field has supposed for the last 30 years. So studies on optic flow, which is the perceived motion that an observer has as they are moving relative to that object or surface, has been studied in electrophysiological laboratories looking at area MT. And this actually suggests that maybe those studies should be revisited using more appropriate stimuli with ecological validity, such as this.
Perception and psychophysics. Research areas: real-world natural scenes, object face processing, motion, depth, spatial, and texture perception, color constancy, visual field crowding, visual attention search, perceptual learning, perceptual development, methods, behavioral, computational, neuroimaging. Later, arrows appear pointing to motion, depth, spatial, and texture perception, and visual attention search.
Okay, so I've given you a bit of a taste of some research that we funded in the Perception and Psychophysics program just on real world and natural scenes, but that doesn't really reflect the breadth of the program. There are a number of different areas that people are pursuing using a number of different methods, Although I will highlight, as you probably noticed, even though these people were captured by trying to apply psychophysics to studying real world natural scenes, they also embedded a few other, areas of interest. So looking at depth, looking at motion, looking at visual attention and search. And so more and more we're seeing projects coming in that combine these different areas, looking at them, as more complex scenarios, but also combining different methods.
Perceptual disorder, refractive error.
So I promised you I would, cover the normal visual function and how it's being studied by researchers in the program. But now I'm moving to perceptual disorder and in particular refractive error.
Image of teacher at blackboard, back of student with hand raised towards the viewer. Myopia, far objects appear blurry while those up close are clear.
So to understand what refractive error is, I'm actually going to put you in the point of view of a 6-year-old in the first grade, maybe the first week of class. And if you are a student who has myopia, and this is the age when generally it's initially diagnosed, the objects that are close by, like that annoying student in front of you raising their hand, that's nice and crisp and clear. But the lesson on the board is not. It appears blurry.
Image of hand holding a book. Hyperopia, difficulty seeing close objects clearly.
And then the converse is for hyperopia. If you are a student, you may have difficulty seeing objects that are close, like the words in the book that you're trying to learn how to read.
Text to right of images. Myopia is negative 0.5 diopters. High myopia is less than or equal to negative 6 diopters.
And so, as I've insinuated with this example, there's a cascade of problems that can come from uncorrected refractive error. The educational prospects can be severely impacted. Now, fortunately, it's relatively straightforward to correct with spectacles or contact lenses or surgery. But despite that, uncorrected refractive errors is the leading cause of visual impairment worldwide. In terms of close to home, roughly 40% of Americans have myopia. 4% have high myopia, myself included. And this latter group actually is particularly vulnerable to pathological ocular conditions such as retinal detachments, macular degeneration, glaucoma, that are blinding. So it's really a serious issue.
Myopia public health epidemic. Plot showing number of people in millions as a function of time. Myopia rises from 1,500 in year 2000 to approximately 5,000 by 2050. High myopia rises from approximately 50 in 2000 to approximately 1,000 by 2050. Later, an arrow appears pointing towards year 2020, labeled at 2.56 billion, and a second pointing towards 2050, labeled 4.76 billion. Reference Holden et al., 2016, Global Prevalence of Myopia and High Myopia and Temporal Trends from 2000 through 2050, Ophthalmology.
On top of that, the incidence of myopia has been becoming more and more common over time. And so currently, next year, the global number of people with myopia is going to be about 2.5 billion, but projecting to the year 2050 will almost double.
Table labeled Prevalence of Myopia Estimated for Each Global Burden of Disease Region 2020 and 2050. Each region includes first a prevalence in percent for 2020 and then for 2050. Andean Latin America, 28.1, 50.7. Asia Pacific High Income, 53.4, 66.4. Australia, 36.0, 55.1. Caribbean, 29.0, 51.7. Central Africa, 9.8, 27.9. Central Asia, 24.3, 47.4. Central Europe, 34.6, 54.1. Central Latin America, 34.2, 54.9. East Africa, 8.4, 22.7. East Asia, 51.6, 65.3. Eastern Europe, 32.2, 50.4. North Africa and Middle East, 30.5, 52.2. Highlighted in red and bold, North America, high income, 42.1, 58.4. Oceania, 9.1, 23.8. South Asia, 28.6, 53.0. Southeast Asia, 46.1, 62.0. Southern Africa, 12.1, 30.2. Southern Latin America, 32.4, 53.4. Tropical Latin America, 27.7, 50.7. West Africa, 9.6, 26.8. Western Europe, 36.7, 56.2. Global, 33.9, 49.8.
And just to, again, look at how that prevalence maps out closer to home, shown in red here, you can see the 2020 prevalence level, about 42% for North America, including the United States, of course, but that's expected to be upward over 58% by 2050. I will say this table is showing the estimations of myopia growth in different regions, and you will probably notice regions such as Asia, different parts of Asia, the numbers are even higher. So reaching epidemic proportions.
Myopia underlying mechanism. Drawing of normal spherical shaped eye with a ray of light focused perfectly on the retina at the back of the eye. Eye development. Emetropia mechanism. Eye growth to ideal refractive state from infancy through school age to late teens.
So it's an important health issue. So it's really important for NEI to do what NEI does best, and that's to support work to understand the mechanisms. So part of that is actually understanding normal eye development. We're all born with hyperopia, and during infancy, our eyes experience very rapid growth. It starts to slow down, but continue during school age. By late teens, your eye should be emetropic. That is, it's at the ideal refractive state so that light comes back to a point to land right on the retina perfectly. And this growth involves a regulated coordination of the lens curvature and the axial length. That's when it goes right.
Drawing of stretched eye. Light no longer focuses on the retina, but slightly in front of it, widening back out before hitting the retina.
But for some, like me, and a few other folks in the audience wearing glasses, there's a myopic shift. So that growth becomes accelerated and you get an excessive axial elongation. And there are a number of risk factors that have been noted that the trait runs in families, so there does seem to be a genetic component. There's been an association for the time spent on near work and a higher incidence of myopia. And it does appear that time outdoors may have a protective factor for developing myopia. But, you know, these are relatively broad descriptions, each one containing a number of confounds. So the best way, of course, to really understand is to look at the mechanisms of onset and progression.
Myopia experimental models. Visual experience, environment, affects refractive development. Simple drawing of normal eye with light focused on retina. Block of heavy black and white stripes. Normal vision, bars appear sharp, high contrast.
And so it's been very, very well established, lots of studies, that we do require visual experience for refractive development. And so experience with high pattern, high contrast stimuli such as this gives meaningful information to the eye about its refractive state.
Form deprivation myopia, FDM, animal models. Drawing of eye with gray rectangle in front. Below, blurry image of fuzzy gray and black stripes. Diffuser goggle, loss of detail, loss of contrast.
But if you lose that detail or you lose that contrast, then you can develop a form deprivation myopia. The retina loses that information. And this finding has actually been used to develop a very reliable and widely used animal model of myopia. We can have one eye receiving normal vision, but the other one is fitted with a translucent diffuser goggle so that they lose the detail in that eye.
Graph labeled form-deprived chicks, showing percentage of eye as function of ametropia, measured in diopters. Normal eyes, distribution peaking around +2 diopters. Deprived eyes, distribution peaking around -25 diopters. Adapted from Gottlieb, MD, et al., 1987.
And then you can see in this example with form-deprived chicks, the normal eye has a intact refractive state, but the one with the diffuser goggle develops myopia. And this model cuts across many species. From zebrafish to non-human primates and actually to humans, we all show this form deprivation myopia. And that's really useful because you can develop these different animal models. They each have unique experimental advantages to use them to study myopia.
Table showing in order: animal model, high cell density region, unique experimental advantage, positives and negatives. Zebrafish, area centralis, genetics, fast development, large repository of gene-targeted mutants, but different visual environment. Chick, area centralis, well-established, rapid eye growth, good optics and acuity, well-studied and sensitive to control of refractive state, practical, easily bred, and good temperament, but double-layered sclera and nictitating membrane. Mouse, visual streak, gene environment, breed large litters year-round, fast ocular growth, gene-targeted strains, and easily obtained, but small eyes and poor optics, slow change in FDM. Guinea pig, visual streak, mammal, large eyes, sensitive to control of refractive state, breed year-round, good temperament, but slow growth. Tree shrew, area centralis, close relation to primates, closely related to primates and reasonably fast ocular growth, but complex handling and breeding. Non-human primates such as marmoset and macaque, fovea, human similarities, close relation to human, excellent visual acuity and good optics, but low availability and reproduction rate and slow growth.
And of course, each one has positives and negatives, ranging from appropriate anatomy to what you're looking at to practical reasons on when they breed or how difficult they are to handle. These all need to be considered for researchers. But I want to emphasize that all of these models are represented in the myopia program.
Myopia experimental models, risk factors, and therapies.
And a number of researchers have actually used them, inspired by some of those risk factors that I mentioned earlier, to try to get at what is it about those risk factors that may affect whether you develop myopia or not. And so Terry Young at the University of Wisconsin is harvesting the advantage of the zebrafish model so she can ask how genetics might play a role. So she's combining genome-wide linkage studies on large multi-generational families who have high myopia and is combining that with mutant zebrafish functional studies. When she gets those candidate genes, can knock them out to see how that affects myopia development. Deb Nicola at the New England College of Optometry is harnessing the advantage of a chick model to address that risk factor of how time outdoors might play a role in myopia development. The advantage of the chick model is it's very responsive, it's quick to the light environment, and it's also diurnal. And so what Deb's interested in is not just light, but those light cycles and how they can affect retinal rhythms, particularly related to dopamine release by the retina, and how that affects axial growth in the sclera. Chris Wiltzuet at UC Berkeley has a long history of working with the chick model, but she's now harnessing the advantage of the guinea pig model to ask how therapies might work. And so the nice thing about using the guinea pig, a mammal, is that she can actually mirror the same type of administration protocol and dosing for atropine interventions, and to look to see how that mechanism ends up working. We can also harness the advantage of the primate model in human data, and rather than trying to give any sort of overview on that, I'm going to say that we can ask Earl Smith because we're very, very lucky to have him here. I'm going to say in terms of question and answer, because Earl really follows up nicely, diving right into the myopia research, we're going to have him come up, give his presentation, and then at the end, if people want to have questions, the three of us will be available.